I'm Logan Spector, and an epidemiologist in the Department of Pediatrics. And it's my pleasure to welcome Dr. Maria Monica Gramatges from Texas Children's and Baylor College of Medicine. So I've actually known Monica for quite a while. We overlapped at undergrad at William & Mary, where we were both biology majors. Proud to say. It's a very successful department. Obviously, we got two of us in pediatric oncology now. And then she went straight to medical school at University of Texas, Houston. Is that right? Okay. I'm doing it from memory at this point. Residency at Oregon, where she became friends with Dr. Wes Miller here, the younger, and then fellowship at Stanford. Followed by assistant professor at Baylor College of Medicine, where she also earned a PhD in clinical research. And she's really carved out a nice niche for herself, looking at molecular and genetic predictors of late effects and toxicities in pediatric cancer. Recently was awarded her first R01 on telomere maintenance defects and second thyroid cancer, which is wonderful. And when the medical science training program said that they were looking for women in science to speak in a seminar series of theirs, I thought immediately of Monica. And so here she is, presented with funding from the MSTP Women in Science program, as well as the Children's Cancer Research Fund. So take it away.
And thank you for the invitation to come here. I'm very pleased to be here today and to talk to you guys a little bit about my research. I have gotten a lot of questions about Texas Children's and Baylor and Houston in the past 24 hours because, as you all know, we had horrible flooding there recently. So I thought I'd show a picture of what Buffalo Bayou looked like before and after the flooding in August of this year. It was pretty amazing. I've also gotten some questions about our cancer center. So just so everyone has kind of an idea, we're a pretty large cancer center in the U.S. We had about 800 new cancer diagnoses every year at our center. A lot more hematologic diagnoses. We have a huge hematology center as well. 140 physicians and scientists on faculty, so it's pretty gigantic. We sub-specialize, so I exclusively see pediatric leukemia patients, and that's pretty much all that I do clinically. And then we have 42 research laboratories. So it's a pretty massive operation. We were impacted pretty heavily by the flooding. We shut down our cancer center for about a week and a half. The hospital was open, but the clinics were not seeing patients. So it was a pretty heroic effort to get all those patients seen and back on schedule once we were open again. I'm going to talk to you guys a little bit today about my research, specifically in telomere maintenance defects and risk for leukemia and leukemia therapy-related toxicities. I'll talk a little bit about late effects, too, at the end. An outline of my talk, I'm going to talk about outcomes associated with telomere maintenance defects, host factors that are related to risk for therapy-related toxicities in pediatric AML, and host factors related to risk for late effects of acute leukemias. So I'm going to give you some background on telomeres and what they are, just in case people aren't familiar with this. So my lab studies host genetic determinants of cancer risk and risk for therapy-related toxicities and research that extends from primary hematologic malignancies to late effects such as thyroid cancer. And much of that work that we do rests on the hypothesis that defects in telomere maintenance correspond with cellular aging and may reflect some of the accelerated aging-related effects of cancer therapy. So telomeres are the conserved repetitive DNA protein repeats that are at chromosome ends. Elizabeth Blackburn won the Nobel Prize in 2009 for her co-discovery of telomerase, which is the enzyme complex that extends and maintains telomeres. And telomere length shortens with age due to the end replication problem, which refers to the lagging strand's inability to replicate the last 25 to 200 nucleotides at the 5' end of the lagging strand. So with shortening, once a critically short length is reached for telomeres, that's known as the Hayflick limit, the cells can no longer divide and undergo senescence or apoptosis. So the consequence of this progressive shortening is an overall loss of telomere DNA with increasing age. And that's shown here in this figure, which depicts telomere flowfish, which is a flow cytometry-based technique where you can sort out white blood cell populations and look at telomere length based on age and years. And you can see here in granulocytes and lymphocytes that there's an overall shortening with increasing age. That particularly is notable in early life and in old age, with some stability in middle age. And this graph is based on a population of about 800 individuals ages 0 to 100. And this technique has been used to extrapolate population-based curves, which I'll show you a little bit later, to help us interpret the percentile of telomere length for peripheral blood compared to a general population. So telomere length is largely determined by both hereditary and epigenetic factors, and there are also environmental factors that can accelerate rates of attrition. So there have been numerous studies now that have shown factors like stress and anxiety can shorten your telomeres more rapidly. Psychological stressors. There's been a lot of studies on smoking as shortening telomere length. And this is peripheral blood telomere length, which is used as a proxy for global telomere length in your body. And then lastly, exposures to DNA damaging agents like chemotherapy or radiation have been shown to shorten telomere length. So there's a syndrome called dyskeratosis congenita that is kind of the prototypic example of people who are born with a telomere biology disorder. So persons with this disorder have very short telomeres, less than the first percentile for their age group. And they're typically associated with the clinical phenotype of mucocutaneous triad of oral leukoplakia, skin pigmentation changes, and nail changes, and also bone marrow failure in up to 90% of cases. They also, and related disorders can include liver and lung fibrosis, which are also part of the telomere biology disorder spectrum. And those figures there are examples of the oral leukoplakia and nail changes seen in dyskeratosis congenita. And I'm interested in this specifically because individuals with dyskeratosis have a 40% cumulative incidence of cancer by age 50. And that includes a 200-fold risk for AML and a 2,500-fold increased risk for myelodysplastic syndrome. So these individuals have a very high risk for cancer, particularly AML and MDS. And there are a set of known mutations in an ever-growing number of genes, I think now it's up to 12, that have been shown to reduce telomerase activity and lead to telomere shortening, as well as a clinical phenotype. And those mutations account for about 60-70% of all cases of DC. But in some cases, we see a clinical phenotype in the setting of a very unknown significance, and we have to figure out what to do with that information. So to illustrate this, I'm going to talk about a case where this specifically occurred, and we had a case of a patient who had a fairly significant clinical phenotype. And you can see here on the MRI, he had a cerebellar hypoplasia, as well as an early onset of bone marrow failure and neurologic concerns, as well as the mucocutaneous triad characteristic of DC. He also had an interesting family history with a sibling who had also passed from this, similar features, and parents who were consanguineous, and a remote history and grandparents of low white blood cell count and cancer. So in this case, when the patient was tested for telomere length, this figure here is what the telomere flowfish looks like, and you can see that this patient, shown in the blue diamond, had a remarkably short telomere length for age. So what this figure is depicting, the red line is the first percentile for age, and the blue line is the 99th percentile, and in between is the 25th, 50th, and 75th percentile for age. And this is, again, a population-based curve that comes from a company in Vancouver, BC, that does clinical and research-based testing for telomere flowfish for patients with telomere biology disorders all over the country. But in this particular case, the parents had very short telomeres, but the patient had extremely short telomeres. That prompted sequencing analysis, which uncovered a variant of unknown significance in the TERT motif. So it was a variant that had not been previously associated with any telomere biology disorder. But clearly the patient would have clinical phenotype, so it begged the question of what was the impact of this variant, and did it have a clinical significance? Could we attribute the clinical phenotype that we saw to this one variant? So to test that, we have a method of actually being able to do that to determine the impact of a variant on telomerase function. And this is a figure depicting the enzyme complex telomerase. It's the telomerase reverse transcriptase and the RNA component that forms the template for telomere DNA. And the functional capacity of telomerase is dependent on two particular pieces. One is telomerase activity, which is the ability of the telomerase to latch on to telomere ends. And then the second is processivity, so the ability of the telomerase to continue to make repeats after initially binding to telomere DNA. And those two components are critical to normal telomerase function. We can measure that function using a direct primer extension assay, which quantifies activity and processivity of the telomerase holoenzyme. And the way that we do that is we can create mutant telomerase by inserting mutants into telomerase reverse transcriptase and compare the impact of those mutations to wild-type telomerase. You can immunoprecipitate the telomerase holoenzyme and then put that into a milieu of nucleotides that include a radio-labeled DGTP. And the telomerase will make telomere products. So after you stop the reaction, you have a series of radio-labeled telomere products that you can then view on a gel and see how the telomerase mutant compares to wild type. So in this figure here, on the far left, you can see what wild type telomerase look like. And what you're looking at are the series of repeats, because the telomere repeats are generated are hexamers. So you initially can get a lengthy repeat for the ones that latched on at the very beginning of the reaction and then continue to make more telomere DNA, versus the shorter repeats that kind of latched on the last 50 seconds of the reaction and only were able to make a few repeats. And the activity is the intensity of all the bands that are kind of cumulative in the lane, and then the processivity is how many small and large repeats that's able to make. So you see the processivity up here in the number of larger repeats that are also made in addition to the smaller repeats. So in the mutant telomerase, you can see here from this particular patient, the activity was comparable, but the processivity was remarkably decreased in the mutant for telomerase. And we also did Western and Northern to prove that there were equal amounts of telomerase in the reaction for both cases. So in this case, this particular mutation we found led to a moderate reduction in telomerase activity and marked reduction in telomerase processivity, and it was the first time this particular mutation in this motif of telomerase was shown to have that functional capacity. So it proves that in cases of variants of unknown significance, we are able to show functional impact of these mutations. And this will become important later as I talk more about work with AML. So in pediatric acute myeloid leukemia, which, as you recall, is one of the diseases associated with underlying defects in telomerase, the factors affecting the risk for this disease are largely unknown. AML is a rare pediatric cancer, and it's diagnosed in about 500 children in the United States every year. And genetic risk is relatively uncommon. It's thought to account for less than 5% of cases. But such patients may benefit from reduced intensity therapies, because with regular intensity therapies, they tend to have a lot of toxicities. The overall survival for AML is about 60% with intensive cytotoxic chemotherapy that may include stem cell transplant. And the mortality associated with the therapy alone ranges from 5 to 10%. So it's a very intensive, difficult therapy, such that the toxicities may be life-threatening. The host factors that predict risk for adverse events of this therapy are also largely unknown. So it's not clear why some patients fall apart with chemotherapy and others seem to sail through. We don't have a good way of predicting that up front when we see these patients. This is just an outline for your reference of what AML therapy looks like. It's composed of either 5 courses of intensive chemotherapy or 3 courses followed by stem cell transplant. And most patients receive all 5 courses, but some cannot receive the 5th and final course due to undue toxicities. So we had a hypothesis that rare or novel germline variants in telomere maintenance genes may be enriched in a pediatric AML-MDS population. And then we also thought that rare variants and short telomeres may predict therapy-related toxicities. So I'm going to talk about this first project. Our underlying telomere maintenance defects enriched in pediatric AML. So we had, at our center, we bank all new diagnoses cancer samples. So we had a bank that included 101 pediatric AML and MDS cases. And then we also have geneticists who are very interested in collecting population controls. So I had a collaborator who had a set of racially and ethnically matched controls. And the reason why this is important is because our population is very heavily weighted toward Hispanic. We have 40 to 50% of our patient population is Hispanic, so we didn't feel comfortable using online databases determining population frequencies of certain variants because those were largely Caucasian-based. So we felt like it was important to have a local set of controls for this. We did Sanger-based sequencing of the 4 genes most commonly mutated in dyskeratosis congenita. And then we also did a blinded retrospective chart review looking for potential features that were consistent with a telomere biology disorder like DC. And what we found in this study were 8 rare or novel telomerase reverse transcriptase variants that resulted in missense changes or deletions in about 9% of our AML and MDS subjects and a smaller proportion of our controls, about 2%. And there was, interestingly, simultaneous to this work, I guess about 2 years ago, more than that, 3-4 years ago, a similar project happened through through the Dutch group where they looked at a population of their AML patients and found no increased incidence of telomerase mutations in their pediatric AML cases compared with healthy controls. So the project that we have taken has not yet been published because since we found so many variants, we have to functionally characterize them, and that has taken some time. So this publication is still kind of waiting in the wings. But what we have shown is that the rare or novel TERT variants that we found are associated with clinical features of DC, specifically skin, nail, and mucosal changes. So if you have one of these rare or novel variants, you're more likely to have skin, nail, or mucosal changes that are consistent with DC. And again, this was done by an investigator who was blinded to the variant status. What we've also shown is that a number of these mutations that we found actually are mutations and that they impact telomerase activity in vitro. So this figure is another telomere direct primer extension assay figure comparing wild type with each of the rare variants that we found. And you can see there's a V713E missense change that was associated with a markedly reduced activity and processivity, and another one at F928L, so the 928 residue of telomerase reverse transcriptase that was also associated with reduced processivity. So we're now looking at the normal activity and processivity mutants to see if they had some kind of other telomerase function impact. If you recall, the direct primary extension assay only examines the activity and processivity of the telomerase holoenzyme, but there are a lot of other proteins that are involved in telomerase function in the cell. So we wanted to look if there first was any evidence of telomere colocalization defects, and we're not able to find any in the mutants that had normal activity and processivity. So you can see here that the telomere DNA is shown in red, and the telomerase is shown here in green. That's the HTR, which is the RNA component of telomerase. And when you look at the colocalization, they look very similar to wild type, at least the mutants do. This is a representative sample of the normal ones. So our next step is to insert these mutant TERT into telomerase deficient fibroblasts and measure their ability to extend telomeres over time. And that experiment is taking a very long time. But that data is really interesting and is in direct conflict with the paper from the Dutch group. So we'll see how that plays out. The second part of the project was to look at telomere length and rare variants in telomere maintenance genes to see if they might impact therapy-related toxicities, similar to what we would observe in a patient with dyskeratosis congenita. And for this, you have to understand that myelosuppression is the most common therapy-related toxicity we see with AML therapy. and that there are patients who have delays of up to or greater than 60 days, so 2 months delay, and that occurs in up to 15% of patients with AML. So each cycle of chemotherapy is given over about 10 days with about a 2 to 3 week recovery period, and it's set to give on a monthly basis, but there are patients who are delayed up to 2 months between courses because they have not recovered their blood counts and cannot go on to receive subsequent therapy. So why is that? There are lots of possible reasons, but we wanted to explore prolonged neutropenia as a significant risk factor, since we know for infection-related outcomes, and knowing that the hematopoietic compartment is particularly sensitive to telomere loss, and that there may be some association with aging-related replicative stress and reduced proliferative potential that might be impacting these people and their ability to recover from chemotherapy. So, to answer this question, we looked at a cohort of AML patients. They were enrolled on a children's oncology group study called AAML 0531, which is a large cohort of patients. We looked at a very small subset of those. We looked at patients specifically who had expected blood count recovery. We had about 62 of those who were less than 1 standard deviation above the mean for time to count recovery, and then those who had delayed count recovery who were greater than 1 standard deviation above the mean for count recovery, to see if there were any differences between those two groups in telomere length and underlying telomere defects. We only looked at patients who got chemotherapy alone, so we excluded those who got transplant because we wanted to know if subsequent chemotherapy cycles worsened their ability to recover. In other words, if short telomeres, our hypothesis was that the shorter your telomeres were, or the more attrition you had, it would be harder and harder for you to recover in subsequent courses of chemotherapy because you would have essentially hematopoietic exhaustion. So what we did was we used samples that were collected at the time point of first remission, and they had to be in remission in order to be included. And then we did quantitative PCR, which is a technique that measures the amplification of telomere DNA compared to the amplification of a single copy gene. And that gives you essentially a telomere content that's a proxy for your telomere length. And we did that only at one time point after the second course of chemotherapy. And what that data showed was that when we took the group as a whole and compared those who were in the first telomere content quartile, or the shortest quartile, to the remaining subjects, those patients had a significantly longer time to ANC, or absolute neutrophil count recovery, than those who had longer telomere length. So this is looking at the probability of absolute neutrophil count recovery and the number of days to absolute neutrophil count recovery. And we showed that for both the 4th and 5th courses of chemotherapy, this time point was particularly prolonged. We also showed that finding was independent of age, which was important because telomere length shortens with age, so we needed to do multivariable analysis and exclude that it might just all be age-related. And then we also looked at, this is a busy slide, but I particularly meant to show you, we were interested in the AML-associated cytogenetics, and whether there were some factors that associated with your ability of time to recover, depending on good or poor prognostic factors with AML. So in acute myeloid leukemia, there's a core binding factor leukemia that includes inversion 16, which is a disease-related cytogenetic change associated with a good prognosis. And our patients with longer telomeres were exclusively associated with our good prognostic AML inversion 16 group, which is kind of interesting. And we also showed that if you had FLT3-ITD, which is a very poor prognostic indicator, you are much more likely to have in the shorter telomere group than in the longer telomere group. So what these factors mean has not been assessed yet in a larger cohort, but it is an interesting thing to follow up on, and I'll explain to you in a second how we're going to do that. And then lastly, just to mention, since I talked about our Hispanic population at Texas Children's and how we're so enriched for this diversity in our population, we were interested in understanding the disparities in outcomes that have been shown to exist between Hispanics and non-Hispanic whites. And we have a prospective study in a separate cohort that has been collecting for years now, looking at the telomere length at the end of first and second induction for AML therapy by flowfish. So this is the flowfish technique that I described earlier, and you can see all the little dots where people plot. And what we've seen is that a quarter of our patients have telomere length that's short, less than the 10th percentile, but that seems to be much more likely among Hispanics than non-Hispanic whites. So what does this mean next? So we're wanting to expand this cohort. So I've developed a collaboration with Richard Appling from CHOP, and he has a genetic study in samples from the newer AML study, AML 1031, and we are now repeating our telomere length analysis in this cohort of over 500 patients, specifically to look at the impact of race and ethnicity, but also to look at the cytogenetic changes and if there's those factors still hold true in a larger cohort of patients. With regard to ethnic disparities, I mentioned, and Dr. Appling, and he has a lot of history investigating these questions. So in previous pediatric AML studies, we've shown that Hispanics have inferior outcomes, as do Black children with AML therapy. And it's interesting to think about in ALL, there have been some studies looking at the impact of adherence and access to care, but in AML, their therapy is delivered entirely inpatient, and they're often diagnosed inpatient and then kept inpatient in most centers. So we think that geographic barriers, access to care, and adherence are probably less likely to impact the outcomes in this particular group. So I think there's much more likelihood for underlying biologic differences among AML patients. There are some interesting features in cytogenetic changes in AML outcomes. So there's a translocation, other core binding factor translocation between chromosomes 8 and 21 that produces the AML1 ETO fusion protein. And that's associated with very good outcomes. So this figure is just showing outcomes of various cytogenetic translocations in AML therapy, showing that this particular translocation is generally thought to be a good prognostic indicator. And interestingly, Hispanics in adult AML studies are more likely to have this translocation. And we've also looked at this in pediatric populations, and it seems like it's more common in Hispanics, but the population size has been too small to really tell. So in two previous pediatric AML studies, CCG 2891 and CCG 2961, the Hispanics had 20% of them had the 821 and 15% versus 11 and 6% in non-Hispanic whites. But those numbers, those differences did not approach statistical significance. Well, they approached, but they did not reach statistical significance. So we looked at this in our center, children with AML diagnosed at Texas Children's over an 18-year period. We had 167 patients, of which 43% were Hispanic. And we also saw that Hispanic children were more likely to have AML with 821. And since we have so many Hispanics, we could actually see that this was a significant difference, which you would think would translate to better outcomes because this is such a great prognostic indicator with 80% overall survival that they should do really well. Well, they don't. So we actually saw no difference in EFS or OS, event-free survival or overall survival between Hispanics and non-Hispanic whites. For all subjects, our overall survival was about 55%, which isn't bad. Typically for AML, it's around 40 to 60%. And we did see that favorable cytogenetics, including those two core binding factor changes, were predictive of improved EFS and overall survival. But we also saw that, there was no difference. The Hispanics didn't do better despite having this, more likely to have this change. But also the Hispanics seemed to have a trend for shorter time to relapse. So we were wondering, but it wasn't significant. So we thought we'd investigate a little bit more why that might be. So we looked at transplant outcomes. And we actually showed that the patients who received chemotherapy alone had no difference in outcomes and seemed to do equally as well, whether you were Hispanic or non-Hispanic white. But those who got transplanted, if you were Hispanic, you had a much poorer overall survival and a much more rapid time to relapse. So the poor outcomes weren't toxicity related, they were the disease coming back and then not being able to recover the patient. This was an interesting result that we published but have not followed up with in terms of the biological explanation for why this might be. And we have a lot of theories. It could be related to the donor choice and donor availability for Hispanic patients versus non-Hispanic whites, but it is not clear. Regarding late effects, to transition a little bit to my current focus of research. So I'm finishing up my work in AML, and I'm now transitioning more to survivorship research. And I want to talk a little bit about that because the field of genetic susceptibility to late effects of cancer therapy is really kind of exploding right now, and there's a lot of really interesting work being done. So as you all know, the incidence of childhood cancer has continued to increase. Mortality has continued to decrease. So the number of childhood cancer survivors in the U.S. is expected to exceed 500,000 by the year 2020. This is a huge population of patients, and they have a lot of problems because they have a lot of late effects related to their cancer therapy. So it's becoming more and more critical that we uncover ways to prevent those late effects or mitigate those late effects because these people are burdened by an incredible amount of chronic health conditions. So as Dr. Robeson and Dr. Hudson have shown now, and others have shown, the childhood cancer survivors have both an excess risk and an advanced onset of morbidities associated with the cancer therapy. 70% of childhood cancer survivors now have a severe, disabling, or life-threatening chronic medical condition, and these increase in frequency and severity over time, conferring a twice-fold mortality risk at 30 years after initial cancer diagnosis compared with non-cancer populations. And genetic susceptibility is thought to contribute to this risk. The most work that's been done in this area is in the area of cardiotoxicity, where we know that patients who receive greater than 300 milligrams per meter squared, which is kind of our threshold for when we start to expect late effects of cardiotoxic meds, that some of those patients don't have any side effects or any late effects related to cardiac outcomes, and that those who receive well below the threshold can have horrible late effects related to their cardiac outcomes. So clearly there's some effect of genetic predisposition to these issues that are important to consider. What's also been shown very well by Carrie Ness at St. Jude is that childhood cancer survivors are at an increased risk for premature accelerated aging, and specifically with frailty. So frailty is an objective measure of physiologic and cognitive decline that can be assessed objectively by aging-related metrics like grip strength, lean muscle mass, DEXA scan. If you meet 2 of the 5 criteria, you are classified as pre-frail and also have an increased mortality risk. And if you meet 3 of the criteria, you're classified as frail. This criteria has been applied to elderly populations for years, but has only very recently been applied to childhood cancer survivor populations. And in Carrie's work, she's shown that frailty is very prevalent among survivors of childhood cancer, such that in a group of childhood cancer survivors who have an average age of 33, they have frailty rates that are similar to a non-cancer population that is 65 years old. So they are like 30 years advanced age compared with non-cancer controls, which is crazy. The genetic and molecular mechanisms of this are completely unexplored. It's like wide open field. We have no idea. There's some very interesting NIH provocative questions related to this, trying to ask or get at what are the underlying mechanisms of this, but it doesn't seem to be obviously related. I think what Carrie's work has shown is that the amount of chemotherapy may be related, female gender, they may be at higher risk, but it's the underlying molecular components are completely unknown. So this is interesting to me because telomeres are a predictor of age. So, and we know that there's an inverse relationship between telomere length and aging-related disease, and an increased risk for primary cancers with short telomeres, and an increased risk for chronic health conditions that characterize elderly populations with short telomeres. Short telomeres in your blood have been associated with dementia, Alzheimer's, pulmonary disorders, cardiovascular disease, all kinds of aging-related problems. Mary Armanios is very well known for describing this. She's a telomere researcher, that many characteristics of telomere maintenance disorders overlap aging-related phenotypes, and she shows that here in a figure from her paper in 2013, that specifically in high turnover compartments we see overlap with hair loss and hair graying and skin changes, immunosenescence, low white blood cell count, increased cancer risk, and also low turnover compartments, specifically the pulmonary fibrosis I talked about, emphysema, liver cirrhosis, and other issues. What are molecular mechanisms of frailty or potential molecular mechanisms of frailty in childhood cancer survivors? We have done some work in support of a grant application to show in unpublished data that childhood cancer survivors have shorter than expected telomeres. So using the telomere flowfish methodology from my current R01 and thyroid thickened cancers, For lymphocytes specifically, we've shown that survivors, 44% of those survivors fall below the 10th percentile for their age, which is pretty dramatic, and 13% of them fall below the first percentile, which, if you recall, if you have telomere length in less than first percentile, that's a clinical trigger for doing sequencing. So there's clearly some work that needs to be followed up here in trying to understand if these people have an underlying genetic predisposition for telomere shortening. And we've also looked at this in relation to frailty. And again, preliminary data that has not been published, but Curiness has helped to look at CCSS data, so childhood cancer survivor study data, in populations that I have already tested for telomere length using the qPCR method, and she's found that those with telomere length in less than 10th percentile are about 2 times more likely to report 2 or more frailty criteria. And I'm not talking about this work today, but the red and black dots refer to with or without thyroid second cancers. And while not significant for lymphocytes, we seem to see a difference in naive T cells. And those who have thyroid second cancers seem to have shorter telomeres, specifically in naive T cells, which may suggest an impaired immune surveillance or ability to detect cancer in that population. But that data I'm not showing today. So in short, our model, we believe that aging-related telomere shortening can lead to replicative senescence and features associated with normal aging. And in cells that have telomerase, that decline might be a little bit slower, but in somatic cells that don't have telomerase or an expressed telomerase, that decline can reach replicative senescence over time. But if you have a chemotherapy or radiation hit sometime early in life or when the cancer exposure, then that attrition rate may decline more precipitously, leading to an earlier manifestation of aging-related disease. So that's the general hypothesis of this work. And we're trying to understand this now with other underlying molecular mechanisms like DNA methylation aging and inflammation. So our proposals that we've submitted include telomere length as well as other markers of aging to see if we can try to understand why this is happening in childhood cancer survivors and if it's something that can be treatable or prevented. So that's all I have. So with that, I'll close and acknowledge members of my lab. I have a very tiny lab. My funding, and then collaborators. So I mentioned Repeat Diagnostics. Geraldine Aubert is our research collaborator there. And then we have a collaborator in Sydney, Tracy Bryan, who does some of the telomere biology work. My own center, my survivorship colleagues, and leukemia research group colleagues, and of course the childhood cancer survivor study that supported a lot of my work. And then my own mentors that include people from my own institution and people from other institutions like Smita Bhatia, who's been a wonderful person for me to work with throughout this project. And with that, I'll take questions. Thank you.
We're going to give our audience in the other locations a chance to ask questions too. And if anybody has a question to ask, they need to signal to Nick that they have one by going to the microphone and let us know if you'd like to ask a question. But I don't see anybody moving right now, so let's take questions here.
So telomere fish seems pretty cool.
Yeah.
Yeah.
And it's really hard to do.
Yeah.
I tried.
Okay. So what I like about it is that you can do it by cell type, whereas the qPCR is on the gamish.
Right.
In your experience, how important is it to do the telomere assays in a cell-specific manner?
So I think it's very important, actually. So my preference for the qPCR would be if you could definitely separate out the white blood cells, but even better, separate out the lymphocytes and do there. So there's good correlation. So in other words, if your telomeres are short in your lymphocytes, they are also likely to be short in other white blood cell populations, short in fibroblasts, short in other cell types. There have been now several papers that have shown strong correlation there, but not the same. So even though overall shorter, yes, but as best as one can to separate out cell types, my preference is to do flowfish if I can. The problem with flowfish is that it requires viable cells. So now when we're doing any project prospectively, we save cells. It can be viably frozen cells, so we always draw a sample and viably freeze cells for the flowfish because I think it's a more accurate assay. The qPCR is attractive because you can use it for the very small amount of DNA, so it takes like 100 nanograms of DNA to do it. and you can do it very inexpensively because cyber is super cheap, and you can do it really quickly. So it's great for larger populations where it's too expensive to do flowfish. So flowfish, we get on a research basis. Clinical testing is about $400 a sample. Research testing is about $250. And if you want to do all 6 white blood cell subsets, it's $550 per patient, which is kind of a lot for a research test. So if that's prohibitively expensive, then qPCR is great, especially if you have a case and control group, because I think it's very useful when you're comparing 2 large populations, or a hypothesis that there's going to be a difference, but it's not very translatable just to say, I don't know what a qPCR telomere content of 1.3 means. It means nothing. It's just a ratio, right? But if you have a case and control group, then you can compare them and say this group is shorter, and then do additional, I would think, further studies to kind of get at which is the white blood cell population that's most important, especially with this thyroid second cancer project that I have. I really wanted to do, the most expensive part of that grant is the telomere flowfish, but I really felt like it was important because I wanted to understand if there were immunological reasons why people develop second cancers. And it's been interesting to see that it's actually naive T cells that seem to be the most important there. And I wouldn't have known that if I hadn't specifically interrogated that subset. So the qPCR that we'd done previously does not enlighten us anyway into what actually is happening. That's a good question.
Other questions?
My name is Wesley. I'm with Pediatric Bone Marrow Transplant here at the University of Minnesota. So first of all, Monica, that was a great presentation. I feel like I learned tons. I feel like this should be requisite viewing for all transplanters, actually. Question for you, though. Like, we obviously know that constitutional chromosomal aberrations can impact like how we treat children with cancers, especially with hematolymphoid malignancies. And I think probably the best example of that is Down syndrome. And that changes the way that we approach the treatment. We also know that with clear genotypic phenotypic correlations of like FA and clearly kids who have known dyskeratosis congenita, we often alter the way that we treat their myeloid malignancies or myeloid disease. Do you think that in your cohort of, I think it was 11 kids, that you found these variants and these TERT variants? Do you think that these are really maybe hypomorphic mutations? And do you think that in the future, I know you're trying to characterize them currently, but do you envision in the future that either a genotype or a phenotype with respect to actual telomere, constitutional telomere assays is going to guide how we approach treatment in patients like this? I
think it's hard. So I thought about this, like how we use this information. And it's a little tricky to think about. The only way I can think of that would make sense is to do telomere flowfish, for example, in remission samples at the end of first induction and show that if they had telomere length that was below the first percentile, consideration should probably be given for transplant. I think for leukemia, it's hard to do upfront testing at diagnosis because their blood is contaminated by leukemia cells. So I think the time point that makes the most sense is when they're in remission. And cancer cells also overexpress telomerase. So if you still have a lot of cancer on board at the end of first cycle of chemotherapy, that could be a problem in interpreting the data. But maybe perhaps even at end of second induction, since they're transplanted after their third cycle, you could get that data back and use it to inform your transplant decisions. We have had, I think the other thing is just awareness about clinical phenotype of telomere biology disorders. I mean, to be perfectly honest, we have a case report that's we're about to submit on a patient with Hodgkin's lymphoma, which is not a classic telomere biology-associated cancer, but who had unusual toxicities with chemotherapy, who had prolonged time to count recovery, didn't have a family history at the time when she was going through therapy, but just had a lot of toxicities that ended up getting to our bone marrow failure doctors or as a consult and had an underlying TERT mutation that was a known mutation associated with DC. And when she got her results back, her telomere length was very short. It was after she completed therapy. So she basically has full-blown DC and had radiation and chemotherapy, which is probably not great. And then her dad got diagnosed with esophageal cancer, which is a cancer associated with DC, shortly thereafter. So their family is now all undergoing testing. But it's just to highlight the importance of being aware. If someone is having major issues with cancer chemotherapy that seems unusually bad, then one should probably think about these disorders, because I think a lot of times the awareness is just not there for testing. And the testing is so easy as a send-out test. It's just a matter of knowing to send it.
And the data that you showed for your positive cohort, so from Texas children that did have TERT polymorphisms, was that constitutional, or was that just?
Yeah, so they're all constitutional mutations. Mm-hmm. So they're all taken from remission time points. So they were all, the samples we collect are from diagnosis and then from remission. So they're actually paired samples. So we showed that they were present in both diagnosis and remission. And the patient with Hodgkin's had an underlying constitutional mutation.
So in your work where you correlated the telomere length to prolonged neutropenia, I'm curious if you also found any correlations between telomere length and either ability to achieve complete remission overall, or particularly ability to achieve a CR after that first round of induction, for instance, on the day 14 marrow.
Yeah, so we haven't looked at that yet. That's one of our missions for the larger data set that I was talking about that we're working on with CHOP. Because with our sample set, it was only 115 patients, so we weren't able, and they were basically all white people. So, because it's children's oncology group, so we couldn't look at ethnic disparities, and we couldn't really look at outcomes because there were just not enough patients in the group to tell any difference. But in the group of 500, then we really hope to explore that further, because I think it would be interesting to see. It might be that they do better. I mean, I don't know. We'll see what the data shows.
This is a follow-up to that question. So there was a time when there was an attempt to develop inhibitors of telomerase as a way to augment effects of cytotoxic chemotherapy. That sort of fell by the wayside, but I'm just wondering what your thoughts are on their prognosis, or is it a predictive marker of benefit from therapy?
So I haven't personally worked, so telmistat, I think, is the telomerase inhibitor. And there are actually studies where it's being used in neuroblastoma and solid tumors that are, I think, maybe still active or still underway. I haven't, theoretically it seems to me to be not a good idea, but I haven't tried to use it. It just seems, stem cell renewal relies on telomerase, so I guess theoretically I wonder about the implications of giving someone a telomerase inhibitor in the long term, because we do need telomerase, and you would think about healthy aging and the reasons why you need to have that. I haven't used it in my work. So I think potentially in a subset of cases, if it were something that were circulating for a short amount of time and didn't have any protracted effect, but I haven't seen the data from the studies that have been done and whether it's been effective or what the long-term outcomes of those patients are. So good question, but I haven't used it personally.
Sort of a related question. So in your data with delay in cycles or prolonged neutropenia, if you analyze the data as to identify the kids who had to have delay in cycles, how many are accounted for by telomerase abnormalities, or what?
So in that data, so in the telomere length and time to recovery data, we looked at underlying defects in telomerase and didn't see a difference in the 4 genes that we looked at. So it's interesting, because I think one of the problems we have with telomere maintenance is there's so many genes involved. So now we're up to 12, but we still only cover about 70% of our understanding of desicatosis congenita population. There's a whole bunch of patients who don't have a clear etiology. For this study, we screened 4 genes, and both groups that were delayed and on time had variants. No one had a variant that was clearly associated with DC that had been previously described as this mutation is bad, and people who have this, or the whole family cohort has desicatosis congenita. So we didn't really have an answer from underlying genetic defects. But I would say that telomere length, even in the absence of since there are so many other factors that influence telomere length, like hereditary factors, that these people, we don't know whether it's their underlying, what they were born with, telomere length that was short, that predisposed them to have this problem of delayed recovery, or whether it's something that the genetic factors related to the chemotherapy they received that caused their telomere length to shorten. But there are certainly a wide range of telomere lengths that we're all born with, that regardless of whether or not we have variants in telomerase or related genes, we just inherit this length from our parents, and there are epigenetic factors that cause us to have short or long telomeres. There's a range of normal, And I would argue that people who are at the lower range of normal, even if there's not a clear genetic defect that causes them to be short, that those people might be at higher risk for therapy-related toxicities with chemotherapy. So I think it's just a population difference that causes them to be at higher risk.
Other questions?
You showed the Texas Children's data. I'm going back to the AML patients with treatment failure, Hispanic
I want you
patients.
to solve the transplant problem.
Well, my question.
We don't know why that is.
Well, so my question for you is, that's sobering, and as I'm thinking through our own patient population here at Minnesota, we obviously don't have like sort of a frequent Hispanic population, but I'm thinking through a lot of relapses, and it's getting the wheel spinning. And my question for you is, you said that the cause of treatment failure was relapse and not transplant-related mortality, right? And that's pretty clear from your curves. Have you guys, the data, does it look like the remission status, the intensity of the chemotherapy used, chemotherapeutic agents used, likelihood of GVL or graft versus leukemia with respect to donor, does all that look like it's sorting out equitably between?
So our transplant center uses a set number of protocols for AML. It's very limited.
Yeah,
there's
exactly.
maybe a handful, maybe one or two protocols that were used to condition these patients all the same.
Yeah.
I suspect it has to do with the limited donor pool for Hispanics because, as we know with the bone marrow registry program, I haven't looked to see, honestly, we want to collaborate with our transplant team to look at this more carefully because they have all the data on the HLA typing.
Yep.
But how many of them received? It didn't look like on my review that there were differences in whether they got a match-related donor or unrelated donor transplant. So that seemed to be the same, and the conditions seemed to be the same. But the donor type and how they selected the donor, I would imagine they probably had fewer to choose from. And I don't know if there were limitations to the donor pool.
But I would maybe hypothesize that they were overrepresented with HLA match siblings because we know that that's generally leads to higher risk. I mean, if you look at relapse risk, and so I don't know if they come from bigger, sibling kinships and cohorts.
Versus leukemia
That's right. That's right. And I would probably focus on that to see because if that looks to be equitably distributed between your two groups, that's an interesting and very sobering finding. So.
Yeah.
Other questions? Okay, thank you very much.